Hello everyone. So very good morning to all. So welcome back to another session in physiology. So we'll start today's session with this wonderful quote. Don't let what you can't do interfere with what you can do. It's a very wonderful quote to begin the day with. So this is going to be about tubular transport mechanisms in renal physiology. A very important topic, a confusing topic. So let's try to learn this as simple as possible from exam point of view. So what is tubular transport mechanisms? See, as you know, the structure of the nephrons, which are the functional units of the kidneys. It has a cup-shaped glomerulus, which is covered by the Bowman's capsule. And then you have the tubule. And what are the parts of the tubule? Look at this diagram. The glomerulus with Bowman's capsule. The first part of the tubule, proximal convoluted tubule. Then it continues as the loop of Henle, and loop of Henle has a descending limb and an ascending limb. This continues as distal tubule. Distal tubule drains into the collecting duct. This is the structure of the nephron. And what we are going to do, we will travel all along this nephron from the glomerulus, starting with the PCT, the loop of Henle, the DCT, and the collecting duct. We will go into each of these areas and we will see what are all the substances which are absorbed, what are all the substances which are secreted, and what substance is not changed. So all these are going to be focused in this topic. So starting from the proximal convoluted tubule, the first part of the tubule. So let's see what are all the changes that are happening in the proximal convoluted tubule. So look at this. So I've just tried to make it as an animation so that it will be easy for you to understand. So please don't miss the video. Please look at this animation. So let's take one part of the proximal convoluted tubule and let's see what are all the changes that are happening. So any cell will have two sides. The side which is facing the lumen, this side is called as the apical membrane, and the other side which is facing the blood is called as basolateral membrane. This is important because the transporters are different in the apical membrane and different in the basolateral membrane. Because once there was a question, which is the sodium transporter present on the basolateral membrane for transport of sodium? Okay, so you need to know there are two membranes, apical membrane and basolateral membrane. The first point is 60% of sodium, chloride, potassium, and water are reabsorbed in the proximal convoluted tubule. See, the 60% is what the value given in Genong. So we'll go with Genong 60%, but also keep in mind there are other textbooks which tell you 60 to 65%, or even 60 to 70%. So all these are correct only. But since Genong tells us 60%, we'll go with 60%. 60% of sodium, chloride, potassium, water. See, important salts. What are the important salts? Sodium, chloride, and potassium. And what is the important solvent? Water. So you find that important salts and the important solvent both are 60% reabsorbed in the proximal convoluted tubule, or even maximum reabsorbed in the PCT. So we'll discuss each of these. So first, let's see how sodium is reabsorbed. Look at this. Sodium glucose co-transporter through which sodium and glucose is transported, and that is named as SGLT2, sodium glucose co-transporter type 2. Sodium amino acid co-transporter, sodium lactate co-transporter, and sodium phosphate co-transporter. So now all this sodium which is reabsorbed through all these channels is accumulated inside the cell. Now how will this sodium leave the cell? So let's look at one more transporter. sodium hydrogen exchanger, through which sodium moves inside and hydrogen is moved into the lumen. Now, all this sodium which is accumulated inside the cell moves out into the blood through sodium potassium ATPase. Yes, so we find that there are several co-transporters and one anti-transporter. This is important because this sodium glucose co-transporter, sodium amino acid, lactate, phosphate, all these are a type of secondary active transport, which is co-transporter. What is co-transport? When sodium and the substance moves in the same direction, it's called as co-transport. But look at sodium hydrogen exchanger. This is different because sodium is moving into the cell in exchange for hydrogen, which is moving out of the cell. That's why it is called as sodium hydrogen exchanger. So this is a type of anti-transporter or counter transport. So this is how 60% of sodium is reabsorbed. And keep in mind, of all these sodium transporters, sodium hydrogen exchanger is only the most important type of transport for sodium reabsorption. So this completes sodium transport. Potassium and chloride. The majority of potassium and chloride are transported in between the cells. Rather than through the cells, it is in between the cells. That is called as paracellular diffusion. See, paracellular means adjacent to the cells. In between the cells, there are gaps in between the cells. So through these, potassium and chloride are transported. That is called paracellular diffusion. What about water? What is that channel which transports water? Yes, it's called as aquaporins. So aquaporin 1 reabsorbs water. On the other side also you have aquaporin 1 through which water moves into the blood. So this is how water is transported. So now you know how 60% of sodium, chloride, potassium and water is reabsorbed. So next let's see. 50% of urea is reabsorbed. This is by simple solvent drag. What is solvent drag? When the solvent water is moving, along with water, urea is also dragged. So by this way, 50% of urea is reabsorbed. 70% of calcium is reabsorbed through proteins called claudin 2. It's a junctional protein, claudin 2. 80% of phosphate is reabsorbed. 90% of bicarbonate is reabsorbed. Now, this is important because there are no direct channels for bicarbonate reabsorption. But still, bicarbonate is reabsorbed. How? Look at this. When bicarbonate is filtered from the glomerulus, it enters into the PCT. It combines with the H plus, which is already there. And from where did you get this H plus? Through sodium hydrogen exchanger. So bicarbonate combines with H plus to form CO2 and water. Now CO2 is a gas. It does not need any transporters. It just simply diffuses into the cell. That is called simple diffusion. So by simple diffusion, CO2 moves into the cell. Inside the cell, just the reverse reactions take place. What is the reverse reaction? Carbon dioxide combines with water to form H plus and bicarbonate. This takes place with the help of enzyme carbonic anhydrase. Hydrogen is again exchanged through the hydrogen sodium exchanger, and bicarbonate moves into the blood through sodium bicarbonate transporter. Now, what if I block this carbonic anhydrase? See, when this enzyme is blocked by drugs, it is called as carbonic anhydrase inhibitors. For what kind of diseases can you use carbonic anhydrase inhibitors? Yes, can you think of the use in pharmacology? Carbonic anhydrase inhibitors, most commonly used as Yes, what is its application in renal physiology? What kind of a drug? Very good, Dr. Aisha. Diuretics. They are called diuretics. See, why are they called as diuretics? Just think. Carbonic anhydrase is that enzyme which helps in this reaction. If you block that enzyme by using a drug, carbonic anhydrase inhibitors, this reaction cannot take place. So when this reaction cannot take place, this H plus cannot be formed. When H plus is not formed, it cannot be exchanged through the sodium hydrogen exchanger. So this exchanger becomes non-functional. It cannot function. So it cannot reabsorb sodium. And I told you, sodium hydrogen exchanger only is the major transport form of sodium. So sodium is not transported. So sodium accumulates in the lumen. Sodium is a cation. It will keep attracting anion chloride. So it attracts anion chloride. Accumulation of sodium chloride, salt in the lumen. What will salt do? Salt will absorb, keep absorbing, or it will retain water. So accumulation of water in the lumen. All this water comes out in urine. This is called diuresis. And the drugs which cause this are called as diuretics. See, that is how carbonic anhydrase inhibitors act as diuretic drugs. 100% glucose, amino acid, and proteins are reabsorbed. So what does this mean? All of the glucose, amino acid, and proteins are reabsorbed in the PCT itself. So glucose, amino acid, and proteins will never cross the PCT. So let's see how glucose is transported. SGLT2 on the apical side. Sodium glucose cotransporter type 2 on the apical side. Glucose is absorbed into the cell. On the other side, through GLUT2, it goes into the blood. So what is GLUT? GLUT is a carrier protein which carries glucose. So what is the difference between SGLT2 and GLUT2? Yes, can you think of this? What is the basic difference? Or what type of a transport is GLUT2? In membrane transport, active transport, passive transport. Nobody knows? Very good, Dr. Hari Venkat. Very good. Facilitated type of diffusion. It is a facilitated diffusion. Very good. Passive type of facilitated diffusion. Repeatedly asked in your exams. See, glucose transport on the apical side by SGLT2, which is a type of secondary active co-transporter. On the basolateral side by GLUT2, which is a type of facilitated type of passive diffusion. So it is active transport on the apical side and passive transport on the basolateral side for glucose reabsorption. So what is the tonicity of the fluid which is going to leave the proximal convoluted tubule? It is isotonic. Why is it isotonic? What do you mean by isotonic? Isotonic means it has same concentration as that of plasma. See, when the glomerulus filters the plasma only flows into the glomerulus and it is getting filtered here. So plasma has a concentration of nearly 290 to 300 milliosmoles per liter. So this only is entering into the glomerulus. It is getting filtered. So here also it is 300 milliosmoles per liter. As it crosses the PCT, salts and water, important salts, sodium, chloride, potassium, important solvent, water, both are 60% reabsorbed, proportionately reabsorbed. Salts, solvent, both are proportionately 60% reabsorbed. So will the concentration of the fluid in the PCT change? No, because solvent and the salts both are proportionately reabsorbed. So the concentration does not change. That is why the fluid which leaves the proximal convoluted tubule is isotonic. It has the same concentration as that of plasma because both salts and solvents are proportionately 60% reabsorbed. Formation of ammonia from glutamine. See, this reaction is important because ammonia, which is formed from glutamine, combines with the H plus, which is already there, which came out through the sodium hydrogen exchanger to form ammonium. What is this reaction called as? Buffering reaction. So what is the role of this ammonia which is formed from glutamine? It helps as a buffer. It neutralizes the H plus. Otherwise, urine will become very acidic. So you need buffers to neutralize the H plus. So already we have one buffer, bicarbonate. Now this is the second buffer, ammonia, which is helping you to neutralize the H plus. Another reaction which happens here is conversion of 5-hydroxycholecalciferol, which is called calcifidiol, into active calcitriol by 25-hydroxy vitamin D3 1-alpha hydroxylase. So this is an important reaction pertaining to vitamin D, which is happening in the proximal convoluted tubule. So this completes the transport mechanisms which take place in the proximal convoluted tubule. See, PCT is a very important part because majority of the transporters and reactions takes place in the PCT only. Okay, so another peculiar feature of the PCT is it has 3 segments, S1, S2 and S3. And you can see that S1 and S2 are convoluted and S3 is a straight segment. So one valid point to remember here is we already know that glucose is 100% reabsorbed in the PCT. So to be more specific, it is 90% reabsorbed in S1 segment and remaining 10% is absorbed in S3 segment. 90% in S1 and remaining 10% in S3. Clinical applications. Carbonic anhydrase inhibitors which act as diuretics. What are the drugs? Dorzolamide, methazolamide, brinzolamide, dichlorphenamide. SGLT2 blockers. Canagliflozin, dapagliflozin and empagliflozin. Sotagliflozin is also an SGLT2 blocker, but it has dual properties. It can block SGLT2 and SGLT1. Osmotic diuretics. See, osmotic diuretics act at the level of PCT. What is osmotic diuretic? What is osmosis? Reabsorption of water is called as osmosis. So what is osmotic diuretic? It blocks the reabsorption of water. So when you give mannitol, mannitol stays in the lumen. It blocks the reabsorption of water. Therefore, all water comes out in urine, which is called diuresis. It will keep attracting water. That's why mannitol is called as osmotic diuretic. So these are the clinical and pharmacological applications at the level of PCT. And what is the use of SGLT2 blockers? What if you block SGLT2? Yes, you are blocking glucose reabsorption. So it can be used in the treatment of diabetes mellitus. Drugs for the treatment. Yes, very good. In the treatment of diabetes mellitus. Next, let's move on to the other segment. Proximal convoluted tubule continues as the descending limb of loop of Henle. The descending limb of loop of Henle is very, very easy to learn because it does not have any channels for salt reabsorption. It has only one channel for water reabsorption, which is aquaporin 1, through which water is reabsorbed. Simple. Only aquaporin 1, water reabsorption. That's all. How much of water reabsorption? Nearly 15% of water reabsorption. This part of the tube is impermeable to salts. Very, very important point. So what will be the concentration of the fluid which leaves the descending limb of loop of Henle? Yes, I want all of you to answer this now. Think and answer. The fluid which came from PCT was isotonic. Now the fluid which leaves the descending limb would be, yes, please think and answer. Very good. Aisha, Pavitra, Sharan, very good. It is impermeable to salts. Only water is reabsorbed. So it will be highly concentrated. That is called as hypertonic. Very good, all of you. Pavitra. So it's hypertonic. So another repeatedly asked concept. Fluid which came from the PCT is isotonic. Fluid which comes out of the descending limb of loop of Henle is hypertonic. Now this hypertonic fluid moves into the ascending limb. So let's see the changes happening in the ascending limb. The first part of the ascending limb is very thin. That's why it's called as the thin ascending limb. And as you know, the fluid which came out of the descending limb was hypertonic. Hypertonic means it is rich in salts. So this fluid which is rich in salts, when it moves into the thin ascending limb, because of the concentration gradient, it is reabsorbed passively because already it is more concentrated. So by passive reabsorption, it is just reabsorbed. You don't need any active transporters because already there is more concentration of the salts there. So it will be passively reabsorbed. The next segment, the thick ascending limb, very, very important segment, the tal segment or the thick ascending limb. 25% of sodium, chloride and potassium reabsorption takes place in the tal segment. And all these three salts are reabsorbed through the same channel. What is that channel? NKCC. NK means sodium, potassium, CC for chloride, chloride. Sodium, potassium, 2 chloride channel. See what happens? Sodium is reabsorbed, potassium is reabsorbed, and this potassium is again recycled through ROMK potassium channels, renal outer medullary potassium channels. So it's recycled back into the lumen. Chloride is reabsorbed, and chloride also goes into the blood through CLC KB chloride channels. So this is how NKCC helps in reabsorption of sodium, potassium to chloride. What if you block this channel? Yes, please answer this. What if you block this channel? What type of drugs are they? NKCC blockers. See, they are acting at the level of loop and they are blocking the salt reabsorption. If they block the salt reabsorption, water also cannot be reabsorbed. So lot of water in urine, which is called diuresis. So now can you say what are they called as? Yes, excellent. Dr. Pavitra, Prakash and Prakalya, very good. They are called as loop diuretics. Loop diuretics. So how do they act as diuretics? They are blocking the reabsorption of sodium, potassium, chloride and chloride. So when salts are not reabsorbed, water is retained, water comes out in urine, that is called as diuresis. What is the tonicity of the fluid which leaves this portion? Yes, can you think and answer? Will it be isotonic? Will it be hypertonic? Or will it be hypotonic? See, this part is impermeable to water. So water is not reabsorbed. Only salts are reabsorbed. So water stays back in the lumen. When water stays back in the lumen, what will happen to the tonicity? Yes, very good, all of you. It is hypotonic because water stays back in the lumen, making the fluid dilute. So it's called hypotonic. And that is why tal segment is called as the diluting segment of the kidney. 90% of bicarbonate reabsorption took place in the PCT. The remaining 10% takes place in the TAL segment. Important clinical application. Loss of function mutation of these channels in the thick ascending limb results in Bartter syndrome. There are 4 types of Bartter syndrome. Type 1 Bartter, mutation of NKCC, sodium, potassium, chloride channel mutation. Type 2 Bartter, ROMK mutation, renal outer medullary potassium channel mutation. Type 3 Bartter, chloride channel mutation, the CLCKB chloride channel mutation. Type 4, Barton mutation. What is Barton? See, Barton is a protein which is important for formation of these channels. So when Barton is mutated, it is called type 4 Bartter. So this is about Bartter syndrome. So this completes the thick ascending limb, very important segment. So to summarize thick ascending limb, one important channel for salt reabsorption, NKCC, and one important disease here, Bartter syndrome, one important diuretic, loop diuretics, the tonicity of the fluid, hypotonic. That's why it is called as diluting segment. What are the loop diuretics? Furosemide, torsemide, bumetanide, ethacrenic acid. So all these act at the level of the loop. Next, let's move on to the next portion of the limb. Distal convoluted tubule. See, the structure of distal convoluted tubule is such that it is divided into early DCT and late DCT. So this is early DCT and this is late DCT. The properties or features are different for early and late DCT. That is why we have to learn early DCT separately and late DCT separately. So first we will learn early DCT. So in early distal convoluted tubule, 7% of sodium chloride reabsorption takes place, and it takes place through a single channel, sodium chloride channel, through which sodium and chloride are reabsorbed. And what is that hormone which enhances the activity of this channel? Very important hormone, aldosterone. From where is aldosterone produced? Yes, which gland produces aldosterone? Yes, it is from the adrenal cortex. And aldosterone is called as salt hormone. So why is it called salt hormone? Because it is helping you in salt reabsorption, sodium chloride reabsorption. That's why it is called as salt hormone. This sodium chloride channel is blocked by thiazide diuretics. Yes, the same mechanism. If you block the reabsorption of salts, water also is retained. Thereby, water comes out in urine. Diuresis. These drugs are called as diuretics. Thiazide diuretics. Another important event that takes place in the early DCT is calcium reabsorption through calbindin D28. Why is it so important? Calcium is reabsorbed in the PCT also. But how come it's very important here? Because this is dependent on parathormone and vitamin D. So PTH and vitamin D dependent calcium reabsorption takes place only in the early DCT. Very, very important point. That is through a protein called calbindin D28. What is the clinical application here? Any loss of function mutation of this sodium chloride channel can result in Gitelman syndrome. In thick ascending limb, it was Bartter syndrome. Here it is Gitelman syndrome. And what are the clinical applications? Pharmacology, thiazide diuretics. What are the thiazide diuretics? Chlorothiazide, hydrochlorothiazide, metolazone. Over to cortical collecting tubule. The late DCT and cortical collecting tubule almost have the same features. So we can learn late DCT and cortical collecting tubule together. So let's learn about the cortical collecting tubule. So the first action of cortical collecting tubule. See, cortical collecting tubule is important because it has many different actions and all of them are important. So one by one we'll do that. What is the first event that takes place in the cortical collecting tubule? With the help of aldosterone, type A intercalated cells. What are these intercalated cells? See, cortical collecting tubule is made up of two types of cells: principal cell or the P cell and intercalated cells or the I cells. Intercalated cells are of two types. I cell is of two types, type A and type B. And how will you remember the functions? A for acid secretion, B for bicarbonate secretion. A for acid secretion, B for bicarbonate secretion. So aldosterone enhances the activity of type A intercalated cell. Therefore, what is the role of aldosterone? It helps you in acid secretion. So the first role of aldosterone is it helps in acid secretion. So when it helps in acid secretion, acid is going to be poured out into the lumen. It is going to be poured out into the urine, thereby acidifying urine. That is called acidification of urine. So the first role of cortical collecting tubule is with the help of aldosterone, it helps you in acidification of urine. Look at the P cell or the principal cell. The second role of cortical collecting tubule is 3% of sodium reabsorption. And how is this reabsorbed? Through channels called ENaC. ENaC means epithelial sodium channel, through which sodium is reabsorbed. And on the other side, through sodium potassium ATPase, sodium moves into the blood. When sodium moves into the blood, in exchange for sodium, potassium enters the cell. Because sodium potassium ATPase, it is a transporter which exchanges 3 sodium for 2 potassium. So when 3 sodium moves out, 2 potassium will move into the cell. Potassium moves into the cell and this potassium is secreted into the lumen. So the third role of cortical collecting tubule is potassium secretion. The activity of this ENaC, epithelial sodium channel, is enhanced by, again, aldosterone, the same hormone. And what if you block this aldosterone? Yes, can you answer this? What type of drugs are they when they block this aldosterone? Yes, aldosterone is blocked by aldosterone antagonists. You can't think of any drug? Aldosterone antagonists? Very good, Dr. Prakalyan, Vishnu Bala. Yes, spironolactone. Very good. Spironolactone and eplerenone. So spironolactone and eplerenone are aldosterone antagonists. ENaC blockers, direct blockers of ENaC, amiloride and triamtrene. So what are they? They are also used as diuretics, but they are called as potassium sparing diuretics. Potassium sparing diuretics. Why potassium sparing? Because when they block aldosterone and epithelial sodium channel, sodium cannot be reabsorbed. When sodium cannot be reabsorbed, potassium cannot be secreted. When potassium is not secreted, potassium stays in the blood itself. So they block or they spare potassium from being secreted into the urine. That's why they are called as potassium sparing diuretics. They will keep the potassium in blood itself. They will spare potassium from being secreted into the urine. So called potassium sparing diuretics. So what is the clinical application here? Any gain of function mutation of this ENaC results in Liddle syndrome. See, what is the difference here? Barter and Gitelman are loss of function mutations, but only Liddle syndrome is a gain of function mutation. See, when you say gain of function, it means that particular channel is functioning more and more. So if ENaC is functioning more and more, it will increase salt reabsorption. So when you have increased salts in your blood, it can result in hypertension. So that is why the most important clinical feature of Liddle syndrome is hypertension. You can have clinical questions based on this. So this completes the cortical collecting tubule. So the three important functions and one more last function of cortical collecting tubule at the level of P cell only. Another new hormone, ADH or vasopressin. From where does this ADH come from? Yes, ADH is produced by which gland? ADS is, yes, as I expected, all of you are saying posterior pituitary. ADH is not produced by posterior pituitary. It is produced by the hypothalamus. It is only stored at the posterior pituitary and released whenever needed. So it's actually a hormone of hypothalamus. Very good, Dr. Hari and Vishnu Bala. Hypothalamus. What is the role of this ADH? So it acts on the cortical collecting tubule on receptors called V2 receptors. V2 receptors. See, when ADH acts on V2 receptor, only when vasopressin attaches to the V2 receptor, this will cause insertion of water channels called aquaporin 2. So when you don't have vasopressin, there is no aquaporin 2. Only when vasopressin attaches to V2 receptor, aquaporin 2 is inserted. And this reabsorbs water. And on the other side, you have aquaporin 3 and 4, through which water moves into the blood. So remember, aquaporin 2 is dependent on ADH or vasopressin. Aquaporin 3 and 4 are not dependent on ADH or vasopressin. Only aquaporin 2 is dependent on vasopressin. So how much of water is reabsorbed like this? Nearly 10% of water reabsorption takes place like this. So to summarize cortical collecting tubule, very important part, listen. So the first function, acidification of urine. How will it acidify urine? Type A intercalated cell, acid secretion with the help of aldosterone. So second function, reabsorption of sodium through ENaC, that is also helped by aldosterone. Third function, potassium secretion through ROMK channels, that is also helped by aldosterone. The fourth function, water reabsorption through V2, so that is helped by vasopressin or ADH. So these are the four important functions of cortical collecting tubule. And also keep in mind, the late DCT and cortical collecting tubule can be learnt together because both of them have the same properties. Let's move on to the next part of the tubule. Medullary collecting tubule. So medullary collecting tubule also has the principal cells, P type of cells. And here also ADH or vasopressin, the same reaction takes place. Vasopressin attaches to V2 receptors. This causes insertion of water channels called aquaporin 2, through which water is reabsorbed. And on the other side, through aquaporin 3 and 4, water moves into the blood. How much of water reabsorption takes place by this method? 4.7 to 5% of water reabsorption. See, remember, regarding water reabsorption, the percentages are different. In PCT, maximum water reabsorption, 60% water reabsorption. And that does not need any hormone. In cortical collecting tubule, 10% of water reabsorption through ADH or vasopressin. In medullary collecting tubule, 4.7% of water reabsorption that also requires ADH and or vasopressin. Another reaction which takes place at the level of medullary collecting tubule is urea transport. So urea is transported through urea transporters called UTA1 and A3. Urea transporter type A1, urea transporter type A3. So this completes the tubular transport mechanisms. So let's quickly summarize the clinical features or the disorders related to the tubule. Barter, Gittelman, Liddle. So Barter, there are 4 types of Barter: type 1, type 2, type 3, type 4. Gittelman, defect in sodium chloride transporter of the DCT. Liddle, gain of function mutation of ENAC. Remember, Barter and Gittelman are loss of function mutations, and only Liddle's is a gain of function mutation. So this is just to summarize. And how will you easily remember this? If you arrange alphabetically, B comes first, then you have G, last you have L. A word about water reabsorption. So we already completed water reabsorption. There are 2 types of water reabsorptions: obligatory and facultative. So what do you mean by the word obligatory? Obligatory means compulsory. So this type of water reabsorption, which takes place in the PCT and the descending limb, is compulsory. Compulsory means it is not dependent on any factor or any hormone. It does not need ADH or vasopressin, aldosterone. It does not need any hormone. It just takes place like that. So compulsorily it will take place. 60% of water reabsorption in the PCT and 15% in the descending limb. They are obligatory, which means compulsory. It is independent of ADH. It is independent of blood osmolality. But what about the facultative water reabsorption, which takes place in the medullary and the cortical collecting tubules? See, facultative means, the word facultative meaning is optional. What is the option here? ADH or vasopressin is the option here. So if you have ADH or vasopressin, this will take place. How much? Around 10 to 14% or 10 to 15%. And if you don't have ADH, this will not take place. That's why it is called facultative. It is optional. What is the option? ADH or vasopressin. In the presence of vasopressin, this will happen. In the absence of vasopressin, this will not happen. Around 10%, or let's say 10 to 14% or 14.7 to be more precise, because 10 in the cortical collecting tubule and 4.7 in the medullary collecting tubule. Around 10 to 14.7% of water reabsorption is dependent on vasopressin or ADH. That's why it is called facultative. And why is it dependent on blood osmolality? See, that is because when will ADH be produced? So let's say this is the blood. Whenever the blood concentration increases, or I can say whenever the blood osmolarity increases, it means blood is becoming very thicker. So quickly ADH will be produced. It will reabsorb water, add it into the blood, and it will dilute the blood. So ADH secretion is dependent on the concentration or the osmolarity of the blood. So that is why facultative reabsorption depends on ADH. ADH in turn depends on blood osmolality. So this is about the 2 types of water reabsorptions that takes place here. Yes, that completes our session of tubular transport mechanisms. So to summarize, remember the proximal convoluted tubule is the most important portion, PCT, because maximum salts and water are reabsorbed in the PCT. That is why the fluid which leaves the PCT is isotonic. And this enters into the descending limb. Descending limb is our friend because there are no channels to remember, only one channel for water reabsorption. So only water reabsorption takes place here. So nothing more to remember there. Then, so that is why only when water is reabsorbed, salt is not reabsorbed. The fluid which leaves the descending limb is highly concentrated, hypertonic. And this fluid enters into the ascending limb. So in ascending limb, the initial portion is thin ascending limb. So thin ascending limb, because of this hypertonic fluid, there is a lot of gradient. So just passive diffusion takes place. Then it moves into the thick ascending limb, very important portion, tal portion of the thick ascending limb. So thick ascending limb has one important channel, NKCC, through which sodium, potassium, chloride, all the salts are reabsorbed. And what is the clinical pharmacological importance here? Yes, loop diuretics act at this level, loop diuretics. And what is the important clinical disease here? Bartter syndrome. Bartter syndrome. Any loss of function mutation results in Bartter syndrome. So now this portion is impermeable to water. It will reabsorb only salts. So what will be the tonicity of the fluid which leaves the segment? Salts are reabsorbed, water is retained. So it is hypotonic. Hypotonic. And this hypotonic fluid moves into the other portions, which is your early DCT and cortical collecting tubule and medullary collecting tubules. Early DCT. Early DCT, only one reaction is taking place, sodium chloride reabsorption. So just sodium and chloride are reabsorbed. And what is the important event taking place in early DCT? Calcium reabsorption. And this calcium reabsorption is dependent on PTH, parathormone, and vitamin D. And from early DCT, it moves into the cortical collecting tubule, cortical collecting tubules. And cortical collecting tubules, remember, we learned about four important functions: acidification of urine, sodium reabsorption, potassium secretion, and also water reabsorption. So all this happens in the cortical collecting tubule. From cortical collecting tubule into the medullary collecting tubule. In medullary collecting tubule, only one reaction taking place. Yes, water reabsorption. So cortical and medullary collecting tubule are dependent on hormones. At the level of cortical collecting tubule, you have aldosterone and vasopressin. At the level of medullary collecting tubule, we have only vasopressin. So this completes the tubular transport mechanisms. So many questions are repeatedly asked from this. Another point in tubular transport is glucose reabsorption. So that is a separate topic with the splay effect. So we'll discuss that as a separate topic on another day. Yes, thanks for watching. So start each day happily. So happy learning to all of you. Any doubts you can post, or if you want me to place any slide, if you have missed any notes, you can post it in the chat box. Yes, thank you all of you. If you are done, we can leave the sessions. Thank you. Yes, Dr. Prakal, yeah. Urea is, urea is reabsorbed in the medullary collecting tubule. It is reabsorbed from the lumen, not into the lumen. It is reabsorbed from the lumen into the interstitium. Urea, see, urea is simple. Urea is in the proximal convoluted tubule by solvent drag. It is dragged along with water. When 60% of water is moving along with water, urea is also dragged. Urea is also reabsorbed. That is called solvent drag. By simple solvent drag in the PCT. And in medullary collecting tubules by urea transporters. Urea transporter type A1 and A3. Yes? Any other doubt? You want any other slides? Okay. Thank you. Glucose transport and splay effect, we'll discuss that separately on another day. Thank you so much. Yes, thank you, Dr. Aisha, Harsha, and Hashwini. Thank you.